discussed kind of what everyone was interested in 2 weeks ago, and a lot of people had expressed that they were depressed and had, mental illnesses themselves, and they were looking at psychedelics as kind of a way of relief. And that's why Aaron is here today. We're going to be talking about the neuroscience of everything that's going on in our brains and how psychedelics can possibly relieve some of these symptoms that we're all feeling today, hopefully relieving them. So, without further ado, Aaron Eisen, he's a very, very, very intelligent, bright man. We are all extremely lucky to be here to hear this today, because Aaron's going to go on to do incredible things within the psychedelic industry of Portland, and we're glad to have him. So give it up, Aaron.
I just want to say I'm extremely lucky to have all of you here today, to take the time out of your day to learn about something that's really important. And Evan, before you sit down, I just wanted to give you this as a thank you for bringing me on.
Oh, how nice!
So these are some pipe cleaner neurons that I'm part of a neuroscience outreach group called Northwest Noggin, and we travel to K-12 public schools. Specifically, we try to reach more underrepresented communities, whether it's more rural schools. We've gone to Native American tribal lands. We've gone to correctional facilities. We've gone to youthless home shelters, and we just try and teach kids about neuroscience. And we like to use art as a good kind of medium to teach the kids about it. So that's why you see all these kind of pipe cleaner neurons up here, and I'm going to be using them a little bit just as some visual aid to this little discussion.
Nice.
And this is an organization that I'm in the process of founding right now called TRIP, which is Truth and Research in Psychedelics. I've paid a lot of money to earn this knowledge, or not really to earn this knowledge, but to obtain this knowledge, whether it is the thousands of dollars I spent on going to my undergraduate college, whether it's the thousands of dollars I spent going to conferences, whether it's the thousands of dollars that I spend to get around all these research articles, because there's all these paywalls to them. I think it's really important to break down these paywalls and to share information freely with other people. So we're going to go over a systematic review of what psychedelics do and how they could be useful in the treatment of depression and PTSD. A title that I kind of wanted to make for this was Systematically Reviewing of Systematic Reviews of Systematic Reviews, but that might have been a little too long for the slide. And we also have some slide credit as well from a few other people. I borrowed as well, so I'm just going to move that up so we get. I move this a little this way. So my name's Aaron. These are just a few of the things that I've been a part of. Now it's a little too high. There we go. Cool. So I'm about to graduate with my Bachelor's of Science from the Honors College at PSU with a dual degree in Social Science and Psychology. I'm a student intern at the Developmental Brain Imaging Lab, where we give 21-year-old participants 1 to 4 substance and then look at their brain connectivity. In fact, I have some flyers up here. If you know anybody who's 21 years old and wants to participate in this study, it's $240, a free picture of your brain, and a few other things. I also work in the ADHD research study, where we're trying to identify different subtypes of ADHD so that therapists could better treat people who have this condition, because a lot of times people with ADHD will have different behaviors than others, so we're really trying to look at that specifically. I also work at the Dornbecker Children's Hospital, the National Registry, where we really try and we're trying to standardize care, so we're at a bunch of sites all across the United States, and we just basically take information about what works, what doesn't work, and then care providers and researchers from all over the country look at that in an effort to standardize care. Also work for DHS as well as a personal support worker. I've just been accepted as a graduate student for integrative medical research at the National University of Naturopathic Medicine. As I mentioned, I work with Northwest Noggin as well. I also know a lot about neuropsychopharmacology, which is what we're going to be covering a lot of here today, as I'm a teacher's assistant for one of my professors. I'm an officer of the Neuroscience Club at PSU, National Honor Society vice president. I will also be receiving Zendo Project peer support training this summer in New York at the Chapel of Sacred Mirrors. I'm very excited for that opportunity. And I'm currently working on a research project in Canada Data, which is the National Consortium on Alcohol and Neurodevelopment in Adolescents. I've been very lucky to have been given this data from the Developmental Brain Imaging Lab. It's across 5 sites across the United States. It takes a lot of different information about adolescents, and the main goal of the study is to kind of see what predicts people using alcohol, specifically adolescents. However, they have just a wealth of information. Specifically, I'm looking at a project right now comparing psychedelic users to non-psychedelic users and comparing their executive functioning. And I found that we have a small group of psychedelic users. There's about 55 of them compared to about 500 non-psychedelic users. However, on measures of behavioral regulation index, which consists of inhibition, so ability to inhibit your behavior, as well as monitor your behavior, the 55 psychedelic users scored higher than the entire 500. And you might say that, okay, like the 500 users, there's more variability. However, according to statistics, central limit theorem, once you have about 30 people, that kind of kicks in, you kind of get this bell curve. So the bell curve was higher for those 55 compared to the 500. And so that was an interesting discovery. I want to make a disclaimer that nothing here represents the opinions of OHSU, PSU, NUNM, DHS, and Canada, or any of the places that I just spoke about. I also just want to kind of say that kind of what I'm going over will be slightly generalized because I don't really have time to go into the full depths of neuroscience. However, we will cover a lot. But if you have any questions and want me to go a little bit further into it, I'd be happy to do that at the end of the Q&A.
Yeah, Aaron, I would also just like to mention, something I forgot in the beginning of this was that I see a lot of familiar faces. I know we're all aware of what Portland Psychedelic Society is. I just need to say that this is a purely educational and informal event. There's no soliciting, exchange of services, exchanges of goods going on within the perimeter.
So now I want to talk about a little bit why I'm interested in psychedelic and psychedelic therapy. I'm currently in recovery from about 8 years, and no conventional treatments were effective for me. I really wanted help, but nothing was working for me. I was at Queens Hospital in Honolulu and Northwest Behavioral Health Services here in Gladstone, Oregon. However, self-medication of psychedelics saved my life, and it really helped me to kind of overcome the difficulties that I was dealing with. So I'm really interested in researching what worked for me in that and understanding the underlying mechanisms so that I can recreate that for other people as well. But enough about me. Let's get to the lecture. So hallucinogens, drugs that produce unusual sensory, perceptual, and cognitive distortions. So there are many different types, plants and synthetic. And there's some good classic examples, mescaline, psilocybin, DMT, LSD. So we have a long history of psychedelic use. It's an entheogen, so it's been used kind of spiritually for thousands of years. And actually, ayahuasca consumption predates the Egyptian pyramids. They found basically old containers that ayahuasca was kind of brewed in all the way prior to the construction of the Egyptian pyramids. And then there's, of course, Terence McKenna's stone ape theory, which is kind of we were consuming what was ever in our environment, which can cause these alterations. And as we're going to learn a little bit later in the lecture, psychedelics actually improve neurogenesis. They actually cause new neurons to grow in certain areas, as well as improve synaptic connections. So it's kind of interesting to think about, well, if we were eating, these things in our environment, how did that influence our development? And there's a really interesting theory about why humans, of all the creatures, all animals on Earth kind of follow a same neuron amount, like the amount of neurons in a specific area. So like on a 1 inch square, the neuron to weight ratio, they kind of follow this trend, except humans. They're the one organism that does not follow this trend. So why does that happen? Like why are we the only species on Earth that kind of broke this trend? And a lot of that has to do with fire. The fact that we're able to our ancestors found fire, they're able to cook our foods, break it down, that form of pre-digestion allowed us to consume more nutrients. Your brain consumes about 20% of the calories that you intake. So that calorie increase allowed our brains to develop more. And then especially if they're consuming substances that would improve that development, that has a lot to do with, or could have a lot to do with how we developed as well. Another interesting thing is animals will not re-administer psychedelics. Like if a cow eats a mushroom in a field, they're not going to go and eat that mushroom again. Humans are the only creatures to re-administer psychedelics, which is very interesting. So kind of more pre-modern, LSD was initially available to psychiatrists in the 1940s. You could just sign up for it and they'd send a bunch to you and you could do some research with it. And then there's two major types of therapy. So psycholytic therapy, which is more popular in Europe, which is kind of the therapy that I would kind of more align with, which is it helps to release the repressed memories. It kind of helps you work with whatever you're dealing with. Compared to psychedelic therapy was originally thought to be as a spiritual thought shock. They kind of like hit rock bottom to kind of go, oh, and pull you out of that. However, we're going to learn it's a little bit more complicated than both of those. And then, of course, the famous MK Ultra experiments where they supposedly used LSD on U.S. citizens just to see what kind of effects they would do. In fact, I was told by one of my professors, and one of their experiments was involved injecting methamphetamine and heroin into both arms at the same time to get this person just to babble incoherently. So it's quite gruesome projects. And there's quite a few others that are not listed as well. In fact, my professor who graduated from OHSU in 2001 said when he was doing his PhD program, which would be in the 90s, he found this article that was talking about LSD experiments on prisoners, where they gave the prisoners LSD every single day for over 70 days. And he was able to find that in like a dark restricted section of the OHSU library. And to this day, we've been looking for any kind of digital copy of that. We have not been able to find it. So if anybody finds that experiment, please send me an email. So your brain is made of cells called neurons, which is kind of like what we have here. And they carry electrical messages and chemical messages. And originally there was the theory of the supers versus the sparkers. And the supers are kind of like chemical soups. And then the sparkers believe that they kind of transferred messages with electricity. And there was kind of this scientific debate for a lot of time. Definitely the supers are kind of winning right now for the fact that we use chemicals called neurotransmitters to communicate. However, up to 20% of the neurons in your brain do not use neurotransmitters. They just communicate through electrical signals directly. So we can kind of see here, there's you have two neurons that will talk to each other. And then these kind of branch-like parts are called dendrites, which is Greek for tree-like branch. And then this long wire is called an axon. So at the end of the axon, you have axon terminals, which are kind of these wires. And they don't actually touch. There's a small little gap between the two. And they'll send messages kind of between each other, which is kind of what you see here. So you have one neuron there, the other neuron there, and those messages are kind of traveling back and forth.
What do they say?
Well, they either say turn on or they say turn off. So they can either send an inhibitory or excitatory signal or send, as we're going to see, there's different types of receptors that can cause like a chemical cascade and cause intercellular changes as well. So all our cells have membranes. So the inside, kind of like the bark of a neuron, has like this phospholipid bilayer. It's these two very strong phospho with like little lipid tails that form together to make walls that are very impenetrable to most substances, except if they're lipids, like cannabinoids. They can like sneak in there fairly well. Most drugs will bind to these receptors, such as hallucinogens, and they cause changes. They cause a change in shape to the receptor, and that change in shape will cause an intercellular cascade and different effects to occur. So just to kind of better explain that, here we have some neurons. So the synapses are where any two neurons will connect to each other. So they're all circled there. However, to see the neurotransmitters, we're going to have to look a little bit closer at it. So here, there's a synapse, which would be probably going to target this one right here. Although we got to look even a little bit closer to see the neurotransmitters. So here you have a synapse. So on this end, you have the axon terminal, and then on this end, you have the dendrite. So chemical messages or electrical messages will travel down, and then this will open small calcium-gated channels that will pull these membrane vesicles down to the docking complex. These little stars that you see in here are neurotransmitters. So once it's pulled down to the docking complex, the neurotransmitters are released into the synaptic cleft, and they bind to little receptors, which then causes the signal to continue going down. So drugs like psychedelics target these receptors. So here we have some psychedelics, or psychedelic substances. They actually target the same receptors, and they'll send either an inhibitory or excitatory message, depending on where it is. And also the environment you're in, which is why hallucinogens, you'll have different trips whenever you take one, because you might be in a different sensory experience, and that really depends on the environment that you're in. Versus drugs more like amphetamine or cocaine, they seem to act on more these specific reward networks. So if you take it, no matter kind of what the environment you're in, you'll kind of get a similar effect. All hallucinogens act on one receptor. That's what makes it a hallucinogen. The 5-HT, which stands for serotonin, 2A, which stands for postsynaptic, which is kind of just learned about. You have the axon terminal and the dendrite. So if it's on the dendrite down here, it's postsynaptic, then the R receptor. And serotonin has about 15 different receptors, so this is one of the 15. We actually see that the better a drug binds to that receptor, the more potent it is. So here you have a little scale, and it says this is more potency, and this is how well it binds to that receptor. So you can see LSD is way down here, so it binds very well to the receptor, thus it's very potent. And I always thought this was interesting because you think of DMT as being one of the most powerful psychedelics. However, you got to think about the dosage of DMT. You're using much less LSD than you were, because about 100 milligrams of DMT is around an active dose versus 100 micrograms of LSD, so that's quite a big difference between the two. So where are these receptors? They're in the neocortex, which would be kind of this primary kind of outer bark around the brain, and that consists of like the frontal lobe, the parietal lobe, and like the occipital lobe back here. So frontal lobe is more decision making. Parietal lobe is like knowing where you are and like knowing how to send movement outward, so motor output. And then down here would be your visual cortex. And then the hippocampus, which we're going to learn is very important for memory. We're going to learn a lot about the hippocampus soon when we start talking about psychopathologies. Your olfactory cortex, which is smell, so the way you kind of smell things and perceive things. And it's very interesting how the olfactory cortex and the hippocampus are kind of linked together. All senses travel through this one part of the brain called the thalamus, which kind of picks up all your senses, except for smell. Smell bypasses the thalamus and goes straight to your hippocampus, which is mostly responsible for memory. And kind of an interesting thing about that is you got to think more primal. How do animals kind of perceive their, threats? They smell them when they're coming. So you got to think back maybe when we were more, primal, like not enough civilization. You probably would smell a saber-toothed tiger before you saw or heard one. So that might be a good time to get out of there. The basal ganglia, which is really important for like finite movement. The thalamus, which I kind of just talked about as well, it's for sensory gating. Cerebellum, which is for more overall movement, although it's, again, that's kind of generalizing, it's quite complex what the cerebellum does. The brainstem, which is a major source of your neurotransmitters and where they're kind of released throughout the rest of the brain, and then the spinal cord, which is inputs. So they cause changes in perception. So these are some cool quotes that I thought were really interesting about people who had taken psychedelics and kind of were saying how it made them feel and how these kind of changes, affected them. Because we can kind of talk about the neuroscience behind it all day, but when you start bringing it into what is it actually, okay, so a psychedelic binds to a receptor, what does that actually cause for our sensual, or, for our actual experience, like for our sensual experience? So hallucinogens affect the gating of sensory input, which is, again, related to the thalamus. So the thalamus sends projections all throughout the cortex, and you can kind of see why that's so important. Here you have the brainstem, and the thalamus kind of sits on top of the brainstem. And you could think of it kind of like the operators for kind of like an old telephone system, where they're kind of like plugging in the different wires to the correct areas. So it's really interesting. Hallucinogens shut the thalamus down, so it's no longer sending information where it should be. And when that happens, you get connections that occur where they necessarily aren't normally. So that's kind of with the thalamus activated. It's sending information to certain areas of the cortex where it should be. And when it's shut down, you just have this like explosion of connections that are occurring. So it really helps to increase cortical regions and gating. And you can see much more connectivity in the brain. So this can, there's major changes in the sense of self and who you are, and just being able to kind of let go of the ego, which is something we're really going to kind of cover in a little bit. And just being able to kind of perceive what your world is around you. And just, a lot of times different psychopathologies will just be overwhelming. Like you have to live with them every single day of your life. And just the ability to step away from that, even if it's for a few hours, can make all the difference. They also activate a network called the locus ceruleus. And the locus ceruleus is a major network of norepinephrine, which is used mostly for attention and orientation. So if you were to walk out into the middle of a road and a car was driving at you, your locus ceruleus would orient you towards the car, or if you were looking at something. Psychedelics hijack this network and orient you to things you might not normally have been oriented to, like your hand or that chair over there. Wow, that chair is really cool all of a sudden. So it really kind of affects where your focus is. And then possibly one of the most important therapeutic aspects of psychedelics is it shuts down the default mode network. The default mode network is kind of your network that you would be thinking about, what am I going to eat tonight? What am I going to do next Tuesday? Like, what do I want to get my degree in later in life? So it's really your network for planning, and psychedelics really shut that down. Like, try and think about what you want to do next Tuesday when you're in the middle of a psychedelic trip. It would be very difficult, which kind of promotes like in the moment thinking and mindfulness, just being able to, realize where you are and in that exact moment in time, which can be very, very beneficial, especially for psychopathology such as PTSD and depression. They're very much kind of built up over time, and being able just to kind of shut that down temporarily can make all the difference. So there's different types of psychedelics. We're mostly going to talk about the classical ones here, which is LSD, DMT, shrooms, so on and so forth. And then there's hallucinogens, which is like MDMA, so that would be, they can kind of have a little bit more mechanisms to them as well. They also affect serotonin, however, they also release things like oxytocin, dopamine, a few other things which can cause a little bit of a different experience. Then we have dissociatives, which they are a little bit more the gnarly side of psychedelics. They can definitely cause depersonalization and kind of derealization, which can be beneficial in some therapeutic settings. In fact, they are looking at ketamine as a treatment for very severe depression. That's a PCP down there as well. They all affect this NMDA receptor and the specific region in your parietal lobe. And it was very interesting. They found that by stimulating this specific region of the parietal lobe, they're actually able to produce an out-of-body experience. So they had somebody with their brain exposed, and you can actually do brain surgery while the person is awake. You don't have pain receptors in your brain. So they were stimulating different networks, and they found when they stimulated this one network, the person reported feeling like they were floating out of their body over and over. So specifically dissociatives seem to target that one specific network. And then we have kind of the darkest psychedelics, which are like deliriants. They're confusion. They're the ones that like people tend to disrobe a lot on. Plucking hair, loss of control, and surprisingly, they're completely legal. And they include like antihistamines, like Benadryl and motion sickness pills as well. They can be very, very intense. So this is on a study that was on a healthy population for LSD, and they wanted to see what basically LSD would cause for long-term changes in the sense of self. And they found some pretty interesting results. They found that it's extremely improved measures of like self-appreciation, life satisfaction, positive social effects, so on and so forth. So it was very beneficial and meaningful for the subjects. And so in a healthy population, we can really help people, and open new doors for people. But what about in people with psychopathology, such as depression, PTSD? How can psychedelics really help them? Because that's definitely a different scenario. So I just kind of want to, before we get into how exactly those psychopathologies affect the brain, I want to talk a little bit about the amygdala, which is in psychology, it's probably one of the most researched parts of the brain related to psychopathologies. So it's an almond-shaped part of the brain that is really responsive to fear. It's kind of the basis of your flight or fight response. So it's known for kind of monitoring more negative emotions. So people with PTSD and depression tend to have much more active amygdalas, kind of responding to those threats a little bit more than they should be, possibly. And then you have the hippocampus, which is the memory part. So it encodes more spatial memories. And another thing about kind of the amygdala versus the hippocampus, the amygdala is more implicit memories versus the hippocampus is more explicit memories. So with the hippocampus, you kind of remember what you were doing in that scenario, and that can be really helpful to kind of calm down negative emotions you're dealing with. Versus the amygdala, you might not know why you're mad. In fact, they've done studies where they found people who have had negative experiences with somebody who looked like a certain way have seen people who with a similar facial makeup or similar face bones, and they kind of instantly don't like them. They don't know why. That's because your amygdala kind of stored that negative information about them and just kind of kept it there. Versus your hippocampus is the kind of the part of your brain that would go, no, I actually don't know that person. It was this other person. So in psychopathologies like PTSD and depression, you kind of have that lack of that hippocampus feedback and that increase the amygdala feedback, which we're going to go into a little bit further. So first I'm going to talk about depression. So it's the leading cause of disability among adults. 16 million Americans last year had a depressive episode, although I'm sure the number is much more that's not reported.
Yeah.
There's many different types of depressive disorders. The most common one we think of is major depressive disorder. There's also persistent, substance use, and then a seasonal affective disorder, which is like Portland rain blues when it's not sun outside and you just kind of get sad. And there's many, many different ones as well. And it's both genetic and epigenetic related.
Epigenetic?
So epigenetic would be environmental changes to your genes. So, for example, long-term stress can cause changes in your genes that will ultimately cause different things to occur. So, depressive disorders are associated with reduced levels of neurotransmitters such as dopamine and serotonin. And I kind of want to quell a common misconception about dopamine. A lot of people think dopamine is the pleasure neurotransmitter. It is not. It is mostly used for movement. And to move, you have to be motivated. For example, if you're drowning and somebody were to throw you a life raft, it's dopamine that's saying, go grab that life raft, go swim over there. And that motivates you to move to go get the life raft. Even in a non-pleasurable situation, you will definitely have dopamine released, which has a lot to do with different psychopathologies. And they found that genetic mutations of serotonin genes reduce resiliency to stressful life events. And in some not-so-pleasant studies, they found that medications that block producing serotonin and other neurotransmitters worsen the mood of depressed patients. So serotonin is a really complex neurotransmitter. Like I mentioned earlier, there's 15 receptors, both ionotropic, which means they bind to a receptor and they cause a channel to open that could let sodium go inside. And sodium is a positive ion that can cause electrical signals to occur when you get enough buildup of a positive charge. Versus metabotropic would be it binds to a receptor and a small little thing called a G protein is released. So a small chunk breaks off the cell inside and then causes more intercellular changes. So more metabotropic drug or receptors can cause more long-term effects than ionotropic. And interesting, 90% of serotonin is synthesized in your digestive tract, and it's a major component of the vagus nerve. And there's a huge link between the gut-brain axis as well. In fact, altered levels of serotonin have been related to diseases such as IBS, cardiovascular disease, and osteoporosis. So how do we commonly treat depressive disorders? SSRIs are one of the most common ways that we use. And they take about 4 to 6 weeks to take effect. And they work for some, However, up to as 20% of people are completely resistant to the medication. And it's a lot more complicated than a single neurotransmitter, as we kind of just walked through a few minutes ago. And another interesting thing about that is SSRIs promote positive moods if you're in a more enriched environment, versus if you're in a more non-enriched environment, it actually worsens your mood. So we got to kind of think about, who's getting treatment, of these people who are getting treatment, how many are in more non-enriched environments that can afford to get treatment, as well. So how does that kind of play into that factor? So there might, people in non-enriched environments might not even be going for treatment. So, and there's so few that are actually resistant to it. I'm sure that this number would be far greater if everybody had the opportunity to receive treatment. And this is just clear evidence that it's far more complicated than simple neurotransmitter imbalance. There's a lot more to it. There's also changes in structure of the brain. For example, less gray matter. And what gray matter is you kind of, again, going back to these neurons, so you'll have the wiring of the neuron, which is the axon, versus you'll have the soma, which is the clump up here. And the soma is the cell body, where it's basically the mission control of the cell. So gray matter would be kind of these clumps of cell bodies up here that are connected, versus the white matter is more the wiring. So you actually have a reduction in the cell body's really important part. In the cingulate cortex, which is like attention, motivation, the hippocampus, which I talked about before, which is specific spatial memory encoding, amygdala, so higher fear response activity. And I thought that was interesting how like less gray matter improved the amygdala's fear response, but you could think less neurons, they might fire more responsively because there's just less that need to be stimulated, versus in a more one with increased cell bodies, there might be more cells that need to actually trigger before you get a negative amygdala response. And then the prefrontal cortex, which is a major component of the prefrontal cortex that's really responsible for like making proper decisions as well. So we can see changes in memory, emotions, decision making, and motivation related to these depressive disorders. And another component is it can negative changes, so it can reduce neuroplasticity. So there's less synapses that are actually occurring, and even decrease neurogenesis. So you have lack of creation of new neurons. The human brain develops to about 24 in females to about 26 in males, and that's generally where neuroplasticity seems to form. However, even in adults, no matter what your age, you kind of seem to have neurogenesis occurring in your hippocampus. So again, this is very targeted towards that hippocampus, and which is really responsible for kind of quelling those negative emotions caused by the amygdala. And then you'll have high levels of cytokines, which are kind of your immune system inflammatory response, which can really promote that kind of like sickness behavior, the lethargy, kind of increased sleep, lack of appetite, all these kind of like negative things that are generally associated with depression. And an interesting thing is more time is spent in bed, but sleep quality is generally decreased. And there's actually no mental disorder that has a normal amount of sleep. That seems to be a characteristic of almost every psychopathology. So sleep is very important. And then there's current measures are like CBT and other psychological measures. About 1/3 patients are treated effectively without SSRIs, just using CBT, which has a lot to say. Just the way you're thinking can kind of cause a change in neuroplasticity on your brain. So it's kind of this ultimate feedback cycle to the neurons are changing your behavior, yet your behavior is changing your neurons at the same time. So depressed emotions can negatively affect the brain regions, kind of what I said, which can create this like recursive feedback cycle, and that can be incredibly challenging to break when you're kind of caught in it, and it might not even be easy to realize you're kind of in that recursive loop cycle until it's far too late to kind of pull out of it. So now we're going to talk a little bit about stress, kind of getting into PTSD a little bit, but I'm just going to talk about stress in a normal situation, just so we can kind of understand kind of the basics behind that. You have different nervous systems in your body. The main one we think of is the CNS. However, we have different sensory and motor ones that kind of feed into the peripheral nervous system. And then kind of the more autonomic nervous system, which we don't really have control over, which is a huge thing in a lot of these psychopathologies, which kind of leads down to the sympathetic division, which is a fight or flight response, which is one thing we're primarily going to be focusing on. And then you have more of the parasympathetic division, so it helps to conserve energy, promotes housekeeping, and it's pretty interesting. Different drugs seem to target certain parts of your nervous system. Like more stimulants seem to target the sympathetic division, kind of stimulating that fight or flight response, while more things like cannabinoids seem to target the parasympathetic division, which is why when people consume cannabis, they can kind of feel relaxed. When you feel like there's no threat, you maybe get hungry, which is why it stimulates appetite, make your nose runny a little bit, things like that could kind of occur. So a major reason why stress occurs, or how stress works in the brain, has a lot to do with the HPA axis, which is the hypothalamic pituitary adrenal axis. And it's responsible for both short-term and long-term stress response. So kind of the short-term stress response would be adrenaline being released, telling you, okay, get out of there. And then the cortisol comes in, trying to stop, inhibit that stronger stress response. Be like, okay, you're out of there, now let's calm you back down. So we kind of have this kind of feedback to it. So it'll basically start at your hypothalamus, and then it sends a message to your pituitary gland, which sends a message to your adrenal glands, which are interestingly located on top of your kidneys. And then your kidneys have your adrenal cortex, and there's kind of the outer bark and an inner bark. So the inner bark, we're going to see releases adrenaline, which causes you to get out of there immediately. And then slower afterwards, the outer bark is going to release cortisol. And the interesting thing about cortisol is we can kind of see that it slowly increases over time. So this might be a stress. You get stressed out, your adrenaline shoots up. The adrenaline is purple, by the way, and the cortisol is yellow. So the adrenaline shoots up, and you got to get out of there. And then at the same time, maybe 20 minutes later, your cortisol starts rising. And then you got out of there, you're okay. So it drops back down. Your adrenaline drops back down. But your cortisol doesn't. Your cortisol keeps kind of flatlining at that slightly elevated thing. Then if you get another stress response, shoots up your adrenaline goes down, but your cortisol will keep rising. So your cortisol will eventually kind of stay on this all upward trend as long as you keep getting spikes in adrenaline, even if you take time to if there's a break between the stressors. So kind of this constant stress will cause cortisol levels to just continue to rise. And cortisol rises about 20 minutes after the stress response. So if you're reading a study and they took cortisol right after they did the stress measure, they're measuring the participant in the car ride before that. They're not actually measuring the participant there. So that's a really important thing if you're looking into research related to that.
Yeah. I just if you grow up in a family where there's chaos and fighting, you're going to have that
absolutely
right there just from the time of you're born up until
Exactly.
And then it doesn't leave, and then you get immune stuff and all these complications and blah,
absolutely
blah, blah.
Yeah. There can be long-term distress has a lot of very, very powerful effects over our body. So I'm going to talk a little bit about the short-term response. So the hypothalamus sends out corticotropin, which then causes the pituitary gland to release adrenocorticotropin. Interesting with the names. I'm glad they weren't too out there with them. So then you'll get that chemical will bind to the adrenal medulla, and it releases adrenaline, dopamine, and norepinephrine, which is a product of dopamine, kind of related to being able to get up and activated and get out of there. So you can kind of see the hypothalamus, sympathetic nervous system, adrenal medulla, and then norepinephrine to get you to get out of the stress. So in the long term, the adrenal medulla releases more things like cortisol, and that kind of prevents a negative feedback, which shuts down the entire process. So good long-term, so that is very important, just being able to remove the stress to continue rising. So like I was saying earlier, great question, too much stress can impact the volume of these structures. So it can increase the volume of the amygdala. Now remember earlier we were talking specifically about gray matter, so increasing the volume of the amygdala could be more more signaling, more white matter, so there's a lot that has to do with that. Lower prefrontal cortex volume and lower hippocampus. And the prefrontal cortex, which I didn't really talk about too much, is kind of the thing that influences this response. So this is kind of a good diagram to see how the HPA axis works. So you'll have the amygdala and the hippocampus, which is really regulating this HPA axis. So the amygdala really helps to turn on the HPA axis, versus the hippocampus is really beneficial at kind of shutting down that response. So if you're looking at psychopathology such as PTSD and depression, where we learned that the amygdala is much more reactive and the hippocampus is less reactive, we can see how this plus would just keep getting bigger versus this minus would get smaller, and thus the stress response would continue going, which would cause a lot of changes.
I have a question.
Yeah.
Back to her comment, for people who have had long-term stress, aren't there negative effects of cortisol?
Yeah, so absolutely. The cortisol is mostly picked. The cortisol doesn't directly cause the effect. It's more, we pull back here. The prefrontal cortex detects high levels of cortisol, and when it detects that high levels of cortisol, it's supposed to shut down the process. Although we can see in certain psychopathologies, it's not effective at doing that. So yes, long-term, just cortisol release would be very, very harmful. In fact, in a few slides, we're going to cover exactly what that can do. So then there's kind of allostasis, which is kind of just improved chemical dysregulation. So all these neurotransmitter imbalances can just kind of increase your stress response. And as we can see here, throughout the life, that can just kind of become overwhelming, even to a point where you don't really realize it's happening, and you're just kind of like stuck under these elephants. So this is kind of answering a little bit of your question. So yeah, absolutely. Trauma, stress can impact a good amount of the body, many different things, including headaches, depression, insomnia, weakened immune system, blood sugar, blood pressure, stomach ache, so on and so forth. There's many different effects that stress has on our body. And I kind of want to make a differentiate between stress versus trauma. So stress is something we might kind of deal with every day, so like a physical, mental, or emotional strain or tension, versus trauma is like too much too quickly, to where you're just not really able to kind of handle that. And then we can kind of see, I think of stress as kind of like tipping the scale a little bit versus being able, homeostasis is handling it and like being able to have it on both. And we can see that that neurochemical imbalance feeding back into your actual emotions can cause that kind of recursive cycle, which can make it very, very hard to achieve homeostasis. So why do we have stress if it's so harmful? So, I mean, this is kind of a cool analogy that I thought of, or that I learned from one of my classes. So you have like a high importance with a high amount of uncertainty, cognitive overload. You're too stressed about something. But if it's not that important and you're not that uncertain, you're not really going to be that interested in it. There's going to be kind of a lack of interest. So optimal performance is kind of this good balance between importance and uncertainty. So it can be important not to have too low, otherwise we just wouldn't really do anything. If nothing was important and nothing was, and we were certain about everything. So now actually getting into PTSD. When does too much stress become actually pathological? So they can be triggered by overwhelming danger, intense feelings, traumatic experience, and it can really affect the fear circuitry, which is kind of what we're learning about earlier in the HPA axis, and it can lead to symptoms that are related to PTSD. So there's acute stress disorder, which is less than 30 days of symptoms, and then at 31 days, it becomes PTSD, once you're still occurring symptoms 30 days later. So, according to the Department of Veteran Affairs, 7 to 8% of the population will suffer from PTSD. I think the number is more than that. And then also experiencing trauma has a commonplace, 60% of men and 50% of women experience trauma. I also believe the number is much higher than that because it's hard to kind of report those figures.
Well, also that's from the Department of Veteran Affairs, so you're not looking at sexual related or childhood trauma.
Absolutely,
That's much different.
Absolutely.
Yeah.
So But it's a little bit more common in women. About 10% develop PTSD compared to 4% in men, and it affects about 7.7 million adults, Americans each year. And about only half of them are actually seeking help for their condition. So there's both, like depression, there's genetic and epigenetic predispositions. So it's generally characterized by an aberration of hormones and the HP axis, kind of like we were talking about a little bit before. And it's specifically related to a certain gene, the NR3C1 epigenetic modifications. And that actually, scientists are finding that PTSD can almost be passed down through generations. So if a mother is experiencing an intense amount of stress, it can cause this epigenetic change, which again, epigenetics is like the stress causes her genes to actually change. Which can,
it happens in the male line too.
Absolutely. Epigenetic happens in both male and female, without a doubt, absolutely. But I was referring to the fact that it can really be transferred onto the...
But I mean semen. It gets transmitted via semen.
Absolutely.
Well, proven.
Absolutely. Well, I did not know that. That's awesome. Or that's not so awesome, but... It's good to know. Yeah. So PTSD, like depression, can cause a reduction in gray matter. So again, in the prefrontal lobe. Interestingly, the occipital lobe, which is responsible for memory and sight. However, the occipital lobe activates when you're also recalling a memory. So if you're thinking about something and you're able to picture it in your head, you're actually using the same neuronal network that you would be if you were looking at something with your eyes open. And then, of course, we have kind of higher amygdala activity, fear response, reduced ability to kind of quell that fear response in the prefrontal cortex, and then again in the hippocampus as well, kind of that same concept of not being able to kind of turn down the amygdala.
I'm sorry, could you say that again?
Yeah, absolutely. So the amygdala, it's really helpful. The prefrontal cortex and the hippocampus are really important at kind of reducing, kind of turning off the amygdala's fear response. So that's a very important component.
About it being the same as if your eyes were...
Oh, you're talking about the occipital lobe. Yes. So they've done studies where they've kind of looked at whether people were just kind of dreaming about something or thinking about something in their head in an MRI, and they found the same areas were activated as being awake and open. Same with hallucinating as well, when you experience hallucinations. In fact, hallucinations generally occur when there's a lack of feedback to a specific area. So kind of what I was saying about the thalamus being shut down before, you kind of have a lack of feedback in certain areas, and your brain will literally draw on similar areas around that to kind of fill in the pieces, which is why you can kind of cause hallucinations. And it's not only visual. For example, people who lose their limbs, they can perceive phantom limb pains. They can still think that their hand is still in pain, and that's directly related to their motor cortex is kind of, there's a bunch of areas that are right next to each other, so lack of feedback to one area, it will connect to similar areas next to it, and then kind of pull back on information like that.
Does that explain to some degree why people with PTSD can feel that they are still in
Absolutely.
that place? It's so vivid. They
And flashbacks.
see exactly everything, can describe.
Yeah.
It's as if they were viewing with their open eyes.
Yes.
What's happening right now?
Right
Exactly.
now.
Exactly.
It's
And in fact, it's when PTSD specifically, those flashbacks are much more intrusive than a thought. Like if I'm thinking of an apple right now, I can easily think of a banana instead. I can switch my thoughts. Versus if you're kind of dealing with a flashback, that can just be overwhelming, and your inability to kind of switch it from another is no longer there. But you're still totally perceiving that, and it's probably a little bit more intense than just seeing an apple, because it's bringing back that kind of amygdala fear activity. When you see that flashback, your amygdala is then, I'm there again. So it's then sending those same messages that you were receiving when you were actually there in the initial thing.
Thank you.
Yeah, absolutely. So again, our friends dopamine and serotonin. Interestingly, increased levels of dopamine, which is related to the fight or fight response, which is kind of related to amygdala reactivity. So you kind of have more stimulation, more necessarily kind of related to the amygdala response. And then you have decreased levels of serotonin, which kind of helps to regulate that relationship between the amygdala and the hippocampus, which is really important in kind of shutting down that negative feedback cycle. And it kind of reduces the efficacy of that feedback cycle, which can increase impulsivity, intrusion of memories, and vigilance as well. So we also have some other important neurotransmitters such as GABA and glutamate. GABAtory is our primary inhibitory neurotransmitter in our brain, and glutamate is our main excitatory one. They're more common than dopamine, serotonin, norepinephrine, and all the other ones. They're pretty much our main signaling in our nervous system. So less GABA decreases the ability to kind of quell our reactions to stress, to kind of calm down that stress reaction, versus glutamate kind of helps to turn on that stress reaction and increases memories and derealization, kind of feeling like you're not like here in the world anymore, like you're kind of pulled back into that experience that you were dealing with. Another one is corticotropin, which is kind of what we were talking about before. Remember, that's the chemical that's released by the hypothalamus to the pituitary gland, which starts the whole HPA axis, as well as norepinephrine. So they have increased activity, which just causes the whole HPA axis to be much more responsive in patients with PTSD. And then this kind of response can lead to numbing, anesthesia, and even disassociation. So just not being able to like feel that you're here, feeling like you're in a completely different place, which is again related to kind of that network in the brain we were talking about before that deliriums and dissociatives kind of activate on. And here's just kind of like a quick overview of all the different areas that PTSD affects and kind of where it affects them in the brain. So we kind of talked about the hippocampus and the gyrus, the amygdala, the prefrontal cortex, the anterior cingulate, and as well as the somatosensory cortex, which is really important for just realizing where you are. Like, for example, if I were to close my eyes, I can still touch my nose right now. And that's the somatosensory cortex, knowing where my body is at this time. And that's really important for kind of motor output as well. So how do we treat them? Again, SSRIs are a very common way that we use to treat them. It's a little more effective, but not that effective than control groups, which is not that exciting to think that this could be a possible treatment that we should really look to as like a savior. And even so in more intense populations, such as veterans, it's not as effective at all. And neither CBT nor a lot of psychological therapies really get at the underlying issues as well as psychedelics possibly could.
Do you know, have there been studies, MRI studies of people with PTSD who have only had the CBT? And have there been much change?
I would assume that there would be a little bit of change, kind of what we were talking about before, that kind of feedback to it, but I don't have the research on that currently. But that would be very something to look into.
I'm curious about your experience using the ACE study.
The ACE study, I'm not that familiar with that.
It stands for Adverse Childhood Experience Study. It's kind of used to, I guess, rate your childhood as far as how traumatic it was. I would recommend it to everyone in here. It's
what was it called?
And obviously it's a questionnaire, so proceed with caution, but I think it's important to kind of understand and know how
it was the ACE? ACE.
And what does that stand for again?
Adverse Childhood Experience. It's used a lot in social work and a lot of the Department of Health Services who work with children.
What about EMDR? Are you talking about that or biofeedback there?
Oh yeah, we're gonna get into the psychedelics in a second, actually.
So
Where's your tongue going? Where's your tongue going?
I think if you're very young and you're
traumatized when you're very young, the brain rewires. It's weird.
Absolutely.
And I can't ever get anybody to understand that my brain doesn't work. You know, so if I'm on somebody's talking to me, or I'm on an SSRI, or whatever those things are called, that's not going to help with the damage in my brain.
No, I mean, it would affect certain parts of the brain, although SSRIs, like I said, we kind of have 15 different receptors for serotonin, so just increasing that, it's kind of has a varied array of effects.
And I've tried every single one of them, and they just create more problems rather than less.
Absolutely, kind of what we were talking about before. Serotonin in less enriched environments can definitely increase or worsen mood and not be as effective compared to more.
Or split you, or whatever, you know.
Absolutely.
So we just there. Do you mean that when they give someone an SSRI, they don't know which serotonin receptor it's going to affect?
Absolutely. It just increases the serotonin in the cleft, and it could bind to any of the 15 ones. So we don't really understand the full mechanism. But yeah, we're about to get into the good stuff. So psychedelics, how can they be used to treat these very, very, very traumatic disorders? We have the 5-HT2A receptor, which we kind of talked about earlier, and it's not just there for psychedelics. It's there for other reasons too, specifically for stress adaptation. So when we're in a stressful situation, it seems that the 5-HT2A receptor is naturally activated, and it actually improves our ability to kind of adapt to a stressful situation. And it promotes neuroplasticity and neurogenesis in the cortex, even in adults. And that kind of neuroplasticity, those changes, can really help you to adapt and deal with a stressful situation. Humans are amazing at adapting to different situations. As we can live in almost any climate in the entire... no other creature could move to any climate in the entire world. And it also stabilizes levels of neurotransmitters as well, which we're going to kind of talk about a little bit more. So they can increase cognitive flexibility, so kind of learning or unlearning of experiences, which we're also going to reference a little bit more. We have improvements in well-being and optimism, kind of these mystical experiences, improve psychological well-being, and decrease depressive symptoms, as well as ego death, which is generally associated with lessening of very strong beliefs that you kind of had before, which can be very, very beneficial to kind of redirect you, or it's kind of hitting like a big reset button. And then kind of dissipation of that recursive loop cycle that I was referencing before, kind of like the ability that neurotransmitters affects your behavior, then your behavior affects your neurotransmitters. So hitting that reset button, if you're in a psychopathological position, can really help to kind of break that negative loop cycle and get you on more of a positive one. So probably one of the most important things they do is something called system regression. So we have low-level association learning. And when we're born, we have these, well, everybody has them, but there are these things called myelin, which are these kind of green, the myelin sheath, which goes down the entire axon. And these help to speed up messages and kind of send messages quickly. So it's interesting, actually, children have less myelin than adults. And that increased myelin in adulthood helps to send messages quicker to where they should be. So it kind of helps with this more projection sense. So you can really kind of plan out your future a little bit better, think about like the long-term aspects. So if you ask, who's a better long-term decision maker, a person in their, this is kind of speaking generally, but a person in like their 30s or a teenager? Probably a person in their 30s might make a little bit better decision because they're able to kind of like send messages a little further and kind of predict that. However, the 16-year-old might see solutions to problems or kind of think about things a little bit differently than the adult would. So there's kind of a benefit to that. So when I'm talking about system regression in psychedelics, it kind of pulls you back to that more younger state. It kind of allows you to think about things a little bit differently, kind of realize things that you might not have, kind of, because it's all about, in adults, it's the best word I can describe it is it really helps to make assumptions. So kind of that system regression removes those assumptions and lets you really think about things as they actually are based on what is available in your environment. And why does the brain work like that? Well, kind of when you're developing, your brain is really gauging the environment you're in, and however you're acting, it's kind of taking that information, storing it, and going, oh, I should act this, or I should respond to X because of Y later on. So that kind of speeding up of messages kind of helps that to occur later in life because you already know what to do in this scenario, so you can respond quickly to things. Versus kids might respond a little slower because they're still learning how to store that memory initially. So a good example, like if you're attacked by a dog when you're really young, you might automatically kind of store a memory that goes, dogs are not my thing, I'm not down with dogs, kind of a thing. So you might not be okay with them later in life. So early traumatic experiences can alter future responses to stimuli. So if you're constantly under stress, then it's kind of causing those changes, which can make you react to stressful situations much differently later in life. And psychedelics can help the brain to relearn these early experiences and alter your responses to future experiences as well. So it's really interesting. SSRIs and psychedelics seem to have do the exact opposite thing. So generally, you have a major component of the psychedelic trip is there's a huge difference between during the trip and after the trip. That seems to be a primary component of the therapeutic mechanisms. For example, when you are during the trip, your amygdala reactivity shoots down. So your amygdala is not as responsive, which decreases your fear response. So you're more open to trying, to work through whatever you're dealing with, or you're more willing to talk about it, just be open about it. And it disrupts the default mode network, which promotes in the moment facilitation of emotions and kind of that mindfulness, not really being able to think about what you want to do 2 weeks from now, just being able to kind of comprehend what you are doing in this exact moment. Interestingly though, higher amygdala reactivity post-trip. So your amygdala does the opposite. Once you're off the trip, it starts becoming more reactive, which can kind of help to which can increase your emotions, but it's found that people are able to kind of accept all of the emotions that they deal with post-trip, whether they are good or bad. SSRIs seem to do the opposite. They just kind of reduce long-term emotional amygdala reactivity, which kind of make this emotional numbness. People kind of describe it as being like a zombie sometimes. So higher amygdala reactivity post-trip helps to improve the psychological facilitation of stressful situations. So having that amygdala reactivity later, actually, with the bottoming out experience initially, can help to improve how you respond to something based on the previous slide of how we kind of can relearn experiences by hitting that reset button with psychedelics. So it causes a greater willingness to accept all emotions post-treatment, even the difficult ones. And they really, SSRI treatments works to kind of the opposite direction, to kind of avoid these negative emotions when you're not really going to help facilitate and get through these experiences if you're avoiding them. Like, they have to be facilitated and dealt with. And psychedelics offer a very unique opportunity to kind of break down barriers that might have been very hard to kind of work and deal through these things without them. And here's some cool quotes that I kind of found from some psychedelic treatments. So I felt a sense of acceptance, more acceptance, agony, boredom, loneliness. So I felt so much lighter, like something had been released. It was an emotional purging. The weight and anxiety of depression had been lifted. So willingness to try and accept the negative times, but also appreciate the wonderful times as well. So this was from a really cool research article by Lisa and Prickett. It was talking about ayahuasca. So they were interested in researching so ayahuasca is effective at treating people with addiction and other psychopathologies, but why? How is it doing that? So they kind of made these four theories, the biochemical, physiological, psychological, and transcendent, which have been kind of bucketed into these two other categories as well. So we're going to talk a little bit about those. So kind of during trip, psychedelics activate the 5-HT2R receptor, they're agonists, and they cause increased GABA in the amygdala, which quells the amygdala, quiets it down. and decreases the utilization rate of monoamines, which is like dopamine, norepinephrine, all the things that we saw higher levels of in like PTSD. And there's decreased GABA in the hippocampus. So it inhibits the dopamine, and that decreased GABA increases the utilization rate of monoamines. So it makes the hippocampus more active. And then it also reduces activity in the default mode network. So it seems to act, the hippocampus has several different parts. It seems to primarily act on the CA areas, which is right over here. You also have the dentate gyrus and another cortex coming back there. But we're mostly going to be focusing on this region of the hippocampus right here, which is really responsible for encoding specific episodes associated with spatial navigation, learning, memory, especially memories of events. Episodic memory, and fact semantic memory, which plays a huge role in PTSD as well as depression. So we have our brain. And kind of in the brainstem, we have the substantia nigra, the raphe, and this is a main center for like neurotransmitters in the brain. So kind of around here, we have our hippocampus, and then down there would be the amygdala. So you have serotonin neurons that project into the cortex, and you have glutamate neurons that kind of project back into the serotonin network. So psychedelics target this specific area down there, and kind of like what we saw earlier, they bind to receptors, and these receptors cause intercellular changes. The 5-HT2A receptor is a metabotropic receptor, so what you just saw here are little G proteins being released that causes intercellular changes. So why does it bind the serotonin receptors so well? So we have serotonin on the screen, and this is DMT. It's a pretty close fit. So we can see that the closer the shape is to serotonin, generally the stronger it will bind, although some chemicals actually are better than activating the receptor than serotonin itself, such as LSD and MDMA. So it then sends a message down back to the neurotransmitter center. And then we're going to kind of take a close-up of the neurotransmitter center. So then you'll have a message, kind of the one we saw earlier, coming down. And it causes glutamate to release its neurotransmitters. And we have a small little GABA interneuron. And then that message will continue on. GABA will be released to two different neurons that will project to the hippocampus. Well, more than two, but just on, I can put that on my screen. But some will project to the hippocampus and some will project to the amygdala. So some will be inhibitory and some will be excitatory. So as we can see here, this is during the trip. The amygdala gets shut down and is less reactive versus the hippocampus is more active. So again, kind of the amygdala being shut down allows your fear response to be reduced, allows you to be more open to facilitating that experience. Then the more active hippocampus helps you to remember how you relearn that experience. And that really helps in future negative emotions or negative episodes. So now we're going to go the psychological, which is more what happens after the trip, kind of more cellular changes. So it occurs through upregulation and downregulation of receptors. So if a cell is getting too much activation, it can reduce the amount of receptors it has. Or if it's getting not enough, it can increase it, which is kind of this upregulation and downregulation, which plays a major key role in addiction. Specifically, you have one network called the nucleus accumbens, which is a major kind of reward network or motivation network, and taking a lot of substances that really activate that, such as like cocaine, amphetamines, can really cause a downregulation in those regions. And that downregulation means that when you have the natural neurotransmitters there's less to bind to, and you'll have less of a kind of a reaction. So like you might not be as happy for waking up in the morning every day because you have less neurotransmitters kind of there, versus when you kind of are working off that, you can cause an upregulation, so that will like improve your motivation to get up in the morning without the substance and just kind of get through the day. So would be during the trip. So then you will see, this would be kind of a lack of activity, so you kind of have lower activation in the amygdala and the hippocampus. And if we want to increase those, high dose of psychedelics promotes learning and memory in the hippocampus. So we can see intercellular changes and more receptors can occur, which causes more places for neurotransmitters to bind to. And thus more reactivity post-trip and improvements in learning and memory. However, if you're trying to promote forgetfulness, kind of delete a memory, you can actually acquire that too from a low dose of psychedelics. So if there's too much stimulation, for example, you're too reactive to a specific memory, too much D proteins are being encoded, psychedelics can help to kind of reduce that so that there's kind of less activity there in the future. And that's with a low dose. So a dose-dependent increase or decrease in the utilization rate of monoamines in the hippocampus, and then we can have more increased utilization rate in the amygdala as well. So you can kind of use that if you want to delete a memory or help promote new learning of new experiences, depending on what the actual treatment you're trying to apply is. We can see that it can be an inhibitory versus an excitatory in the amygdala. So psychedelics enable the processing of significant conscious and unconscious experience, promoting kind of the resolution of the trauma that is being dealt with. And the death of the ego provides this resolution, and kind of this regression and depersonalization is a reparative attempt generally to deal with the internal conflict, which the psychedelics really help to kind of work through. And that's just from one of my other ones. Ayahuasca promotes a oceanic feeling of connectedness to the universe, which resolves psychodynamic issues regarding the environment. Because there can be a lot of different issues. It cannot just be you're not okay with yourself. You can feel you're not connected to your environment around you, whether you're not okay with your sexual orientation, or how your sexual orientation is perceived in your environment, I should say, or if you're not okay with your social economic class. Like, there's all these different factors that can make you feel like not okay with your world around you, even if you're okay with yourself.
I have a question.
Yeah.
You talked about genetic changes caused from trauma.
Mm-hmm.
So then taking the psychedelics for that, can that reverse those changes?
That's what we're seeing. It can actually help to kind of, because those epigenetic changes kind of cause that feedback loop cycle. So taking the psychedelics can help to break that feedback loop cycle. So while taking that one psychedelic won't go into your brain and repair everything right there on the spot, it will start the chemical cascade that will kind of work towards improving that process. If anything, it will prevent further damage from being occurring. Because even if it doesn't, it's hard to say the research around exactly what areas it would kind of cause it to be fully restored or whether it would go back, but it definitely would help to prevent further damage from occurring as well. Yeah.
I've actually read some papers on ayahuasca when I was doing a project in one of my classes, and I actually was reading about it, and they were saying that it kind of rather than just giving you a bunch of serotonin all at once, it kind of slowly but surely, over months, raises serotonin levels in the brain.
Absolutely. In fact, psychedelic therapy is very unique in its mechanisms. They've found that it's the only therapy that they've given to a group of participants one time, measured them after, they all improved. Measured them 1 year later, they improved even more than before. You don't see any kind of therapy like that in our current day and age.
What was the substance on the 1 year?
I believe it was psilocybin.
Do we know why, from a neuroscience point of view, that psychedelics promote a feeling of oneness with the universe?
That is a great question. So they kind of break down that kind of the ego death is a very important component to it, kind of those predispositions of kind of how you are in your environment, kind of breaking those down can really help to do it. But from a neurological standpoint, it really has to do with the 5-HT2R receptor being activated. And remember, the 5-HT2R receptor is responsible for stress adaptation. So kind of having that receptor activated can really help to promote, those changes and being able to kind of adapt to the situation. makes sense. Yeah.
Yeah, I would think it would have, addressing that question, something to do with the default mode network. It being that network that gives us a stable sense of ego, repetitive thought patterns.
Absolutely.
That gets broken down in a psychedelic experience temporarily, which could, lead to, an ego death experience if it was, an intense enough trip. But that sort of circular, I'm me, I'm in this world, this is who I am, this is my role right now, that gets shattered for a while.
Absolutely.
In addition to suppressing the thalamus and allowing a whole lot of different kinds of sensory input to flood in simultaneously.
Absolutely. And different connections to occur, even that are not related to like sensory input as well. But
Yeah, not only generalized depression of the DMN, but also specifically the posterior cingulate cortex that really deals a lot with self-identity, autobiographical memory. It kind of stimulates out of the campus into that region. A lot of that stuff calms down during the psychedelics.
Absolutely.
What did she say?
She was talking about how. Do you want to? Sorry.
The DMN calms down, and that's kind of your self-referencing and ruminating, this rumination part of your brain that does a lot of psychological, self-referencing behavior. And so it calms down that area, but also this region called the posterior cingulate cortex that really deals with a lot of self-identity. That really calms down.
Absolutely. Oh, yeah.
Yeah, so in terms of cognitive flexibility, I'm wondering why the knife doesn't seem to cut both ways, like in terms of like negative moods or positive moods.
Could you rephrase that maybe a little bit?
Yeah, like with cognitive flexibility, like your mind is changing, right? It's able to see things differently. I'm wondering why that it seems to trend towards the positive and instead of trending towards the negative.
Well, you do experience your negative emotions, but it kind of allows you to kind of facilitate those negative emotions a little more. So it's not really just saying you're only going to experience good feelings. It definitely, you still will be experiencing these kind of negative emotions, but it just kind of helps to work through that. And I think that has a lot to do with kind of shutting down the default mode network, causing an ego death to occur, and kind of like neuroplasticity to kind of continue in areas that you are really kind of targeting. Makes sense.
So it's kind of shutting down a subjective part of our brain.
Yeah.
And allows us to be more objective.
Absolutely.
In our experience.
Yeah. So we kind of have this three-horn dilemma, which is this is a major component of psychodynamic theory. So we have the ID, ego, and superego. And the way I like to describe it is like the ID would be two children were playing with a truck. It's one of the child wants to play with the truck, but one is the ID. The ego would be maybe the children are a little older, and the child thinks, if I take the truck and only play with it myself, am I going to get in trouble from stealing the truck from the other person? Like, am I going to get scolded by one of my parents? And then I think of the superego, like when the kid's a little bit older, it's their own morality. Like, am I okay with stealing the truck from the other person? So we generally have a conflict between these. So we want to we can pretty easily satisfy two, but generally at the expense of the other one. So it's the three-horn dilemma. And psychedelics really help to shut down the ego. And that can lead to resolution of internal conflicts. And I do want to kind of differentiate the ego psychologically than the literary definition. Generally, the literary definition is like a bad thing, being like, I'm very caught up in myself, I think highly of myself, versus the ego in a psychological definition is kind of how you interact with your environment. Like, how does my environment necessarily kind of perceive me? How do I perceive myself in my environment? For example, in the scenario I just ran about the child with the truck, they're really thinking, am I going to get in trouble with my mom or my dad if I steal the truck? So they're kind of thinking, what am I going to do is how does that impact the world that I'm in? So that's kind of where the ego comes from. And so the ego death from psychedelics is kind of the shutting down, thinking, I need to be this certain person in my reality. It kind of allows you to be who you actually are and be okay with that. That's how I would describe that. So it kind of dissolves stress. So there's the transcendent hypothesis, which is one of my favorites. So it can change, experiences can change your beliefs, values, and worldview, promoting connections to the environment. And a major component of a lot of psychopathologies in the United States, which is a little higher than other countries, is social preoccupation with individualism. So how we're always focused about, working towards kind of improving ourselves rather than more collectivistic cultures are really about, improving your entire community and your family. Like a good example would be, I grew up 3,000 miles away from here, and if in an individualistic society like we live in, it's not weird at all to move 3,000 miles away to get a good college education. That's very normal. Versus in a collectivist society, you wouldn't see that. You'd see the person kind of living with their family and kind of staying there. And that kind of social preoccupation generally leads to social isolation, which is a huge component of a lot of different mental psychopathologies. We're social creatures. Like, we work by bonding and by communicating with each other and being there.
So would you say that's one of the reasons why usually Americans kind of experience more depression than
Absolutely. And substance use. Like, if you look at substance use in collectivistic cultures versus individualistic cultures, there's a huge difference. Like, if you look at substance use in the United States compared to maybe the Middle East, you're going to see much different results. And that's because, also a very interesting thing I learned was schizophrenia is very different in collectivistic and individualistic cultures. In individualistic cultures, schizophrenia is the FBI is watching me, the TV's talking to me, versus in collectivistic cultures, it's generally I've let somebody down. I brought shame upon myself or my family. It's a completely different fear that they have, which is really interesting, I think. And those kind of social disturbances can pertain to connections in your environment, society, and even your own identity. You feel your gender is not represented fairly, or your sexual orientation, or there's many different ways, or your social economic class. There's lots of different things that can kind of help to, or negatively feel like you're connected to your environment around you, which is very, very important. We can see from a psychological standpoint to kind of feel that connection and feel like you belong where you are.
I'm also kind of curious. So generally, violent people are born with the two short serotonin alleles, so they have that lower stress response, as well as generally a higher level of testosterone, and so they end up being more violent. And I'm just kind of wondering. And then they also people who tend to act on those violent tendencies feel like they're kind of outcasts from society, and I'm just wondering if psychedelics could help that.
I think they could.
Yeah.
I definitely think so. Because psychedelics can really help to kind of bond and have a really good experience. In fact, it was a little controversial, but I was recently at a psilocybin society meeting, and they brought Mark Hayden, who is the director of MAPS Canada down there, and he was giving a very interesting lecture specifically about psychedelic therapy. And one of the topics he brought up was something I've been thinking of for a long time: psychedelic use with adolescents. So, is that something we should consider, research? And his answer was yes. And he asked what was the appropriate age, he said when the child knew what they were asking for, when they were old enough to really comprehend what they were getting out of the experience. And thinking about it, a lot of the times that people start to experiment with drugs and possibly psychedelics is generally kind of in their late teenage years and things like that. And just thinking how a lot of this experimenting is done, generally at friends, with parties, things like that. Just thinking about if we could bring it into a therapeutic setting, even have the parents there, if the parents are open to things like that, have it be an amazing bonding experience for the kids and your parents. That could be a very interesting, although it is a little controversial. I've definitely heard a lot of people kind of say opposing perspectives about that, but it's definitely something that should be kind of researched a little bit more and further, especially considering that with a lot of psychopathologies, especially addiction, catching it while you're young is really important, because not can kind of lead to a lot of things later in life, and things can kind of tend to get more intense. Social connection. So kind of what we were talking about a little bit earlier, and this kind of plays into maybe individuals who are growing up in more traumatic environments, who didn't have as much social connection when they were younger, kind of feeling a gap to that. There actually are significant changes in the brain that occur, specifically the anterior cingulate cortex, the medial frontal gyrus, the temporal gyrus, and several other areas that are very, very important. That are all associated with like euphoria, reward, motivation, and social cognition, even self-representation, so knowing who you are. And here we can actually see that this is from a study of youth who grew up in traumatic environments, and they can see that the red areas are areas that are associated with kind of your neuronal alarm network, so like a threat response, and those areas increased, versus these blue areas, which are more like reward, motivation, they decrease dramatically. So growing up in kind of these traumatic, stressful environments can have quite dramatic changes of the brain. However, kind of like what we learned, psychedelics are an amazing opportunity that can actually improve neurogenesis in adult. While taking this, I want to make it clear, taking the psychedelic once won't just cause everything to be fixed and back to normal. It can definitely start the chemical cascade that will stop harm and hopefully work towards repairing those things as well. And there's definitely good implications from that. And then kind of that lack of social connection, it activates the same physiological alarm network as if you were being physically harmed, not having that connection. And then having that connection acts on the same network that most addictive drugs use, like cocaine and amphetamines as well. That's why when I ever go to public schools, I tell kids, you don't need to use drugs, just be friends with each other. Activate the same with do they listen to me? I don't know. So psychedelic therapy has several different elements to it. This was taken from MAPS. It's kind of the basis of their therapy. So element 1 would just make sure you're in a safe therapeutic mindset. And I do want to say that there is quite a big difference between just taking a psychedelic and psychedelic therapy. There is kind of a gap between there, because psychedelic therapy goes hand in hand with kind of more psychological therapies at the same time. And a major component that I've noticed that's majorly different from common, more psychological therapy compared to psychedelic therapy, in a psychological environment, you'll generally have a therapist sitting in a chair, and you'll have like a table, and you'll have the other person on the other side of the table. That table in the middle instantly causes a gap between you and the person. You just removing that table can really help you just to like connect to that person a lot more. However, in psychedelic therapy, you'll see a lot of the times people are they're wearing like a mask over their eyes, and their hand is being held through the entire trip. Granted, if they want, like if they don't want to be touched, they don't have to be. But that a major component is kind of that connection, like holding the person's hand, which is something we don't see in almost any psychological treatments, like any physical person.
So, are you going to go into more detail? I was just curious.
Yeah, I'll definitely walk through them a little bit.
I don't see how, I mean, somebody has a project they want to work on, you take a drug, and then is there like, well, how are we going to approach, like, even number 2? So are we going to talk about it, like, in a certain way, or they talk about their experience and then go into it, or what does that look like?
So it is in combination with like common treatment. So like kind of exactly what you're saying, like kind of more counseling and things like that, just kind of like working with. So it's basically like an addition to common treatments that we have to kind of improve their efficacy and give the therapist like an extra opportunity. Especially a major component is the exposure therapy, where the reduction of the amygdala allows the person to kind of come out and talk a little bit more about what they're dealing with, and that kind of being able to be exposed to that can really help them to kind of facilitate that as well. But yes, it is in conjunction with basic psychological treatments as well. That is a very component aspect of it. Kind of like I'm saying, if you just take a psychedelic versus take it in a therapeutic setting, it's a very different experience. So, and then, that leads to more cognitive restructuring, kind of what we're talking about, being able to kind of like learn relearn certain associations with different things, as well as working in many different areas of the psyche, and then somatic manifestations of trauma as well. So that would be kind of being really helpful to help the person kind of work through that. So just talking about the current efficacy of current treatments, non-psychedelics is about 10 to 25%. for PTSD. MDMA-assisted therapy has an efficacy of about 83%, which is a huge gap. In fact, when the results were first published, a lot of researchers did not believe they were credible. They were like, no way something could be that effective. And in fact, they have been replicated all throughout the world, and there seems to be, I believe it's kind of a varying between a 60 to 80% efficacy. When you're comparing that to a 10-25%, that's unheard of. Yeah.
How many sessions is that over?
That is a good question. That generally is over 1 to 2 sessions. So just one exposure. It's not like a continuous kind of reback. And kind of what I said earlier, immediately after the session, there was an improvement. But even 1 year later, there was a greater improvement than right after the session. So it was a treatment that causes a long-term kind of cancer. Yeah.
But so how long is a session then?
So
on average?
Generally sessions would be, when I say a session, I mean kind of going through this entire process. Yeah. Okay. So it'd be kind of, and generally they take MDMA about 1 to 2 times throughout this process, which would generally be kind of in this part would be the psychedelic administration, although these kind of components are throughout the entire experience as well. And then MAPS Canada phase 2 trials with a larger and more diverse population at a 60% efficacy. So there's a lot of implications out here, and it continued to increase in life satisfaction 14 months later, which is unlike anything we currently have. Yeah.
With these interventions, is increase in life satisfaction defined as
well, there's a little bit more to it, definitely, but that was like one of the major things because they were dealing with PTSD, so that's a major component is kind of having those flashbacks and just kind of being caught up in that, negative, really kind of difficult situation. A lot of people with extreme PTSD don't report that high of life satisfaction. So they did kind of use that as an efficacy marker, although I'm sure there was more to it than that. But that was one of the most amazing things that was kind of taken from the study, that even a year later, or more than a year later, it increased it. And in fact, the Somatic Center in Portland has just started their MDMA phase 3 trials, which is FDA approved. Yeah.
So there is some trials going on here in Portland.
Yes.
How can we find out about that?
So there is. They hope to start it up soon, but yeah, it's the government shutdown did not help.
I think about 5 months is when they should be starting.
So how do we get involved in that?
So I believe you can, they have a mailing newsletter, and you can sign up for their
newsletter. Is that under Somatic Center Portland if I go there?
Yeah, if you Google them, you should be able to find the newsletter.
Cool. I think the phase 3 trials with MDMA, the phase 1 and 2 also, all are 3 sessions of actually taking the substance.
Okay.
And the efficacy is based on no longer diagnosable.
I see.
So at one point you're diagnosable, you have the DSM, you meet the requirements for diagnosis. And then later you can't be diagnosed with. Doesn't mean you don't have any of the symptoms or effects, but you're not diagnosable as having PTSD, the disorder.
Absolutely. Okay. Yeah, it definitely is not a complete fix-all. It's not like you take it and you're just everything's better. But yeah, I completely see what you say there. There is it's all about kind of getting it to that level, though, like being able to not be diagnosable anymore.
Yeah, I mean, for the people who are suffering,
it's amazing. Absolutely.
I don't understand. Why can't you participate if you do have a diagnosis?
Of PTSD.
Can you repeat the question, please?
Yeah, why can't you participate in these types of trials if you have an actual diagnosis?
No, you can.
You can.
You can, yeah.
Okay.
Aaron, can you repeat the question? Because people back here can't hear.
Sorry about that. She was asking if people with PTSD were allowed to participate in these trials. Yes, they are. These are for people with PTSD. Although, unfortunately, it's currently, because it's not backed by any kind of insurance company, it's out of pocket currently.
Which they are raising money. I think that's part of the trials being approved, is that they have to raise a percentage to provide care for folks who can't afford it. That's part of the beginning of the trials, which is what they're doing now.
So, talking a little bit about psilocybin and LSD, a meta-analysis found that anxiety and depression in cancer patients was reduced as well as symptoms of alcohol and tobacco dependence, which tobacco is essentially one of the most difficult drugs to alleviate or to get somebody off. It even shows higher addictions rates than methamphetamine. Ayahuasca reduced depression symptoms and treatment-resistant depression, and it's also protective against suicidal ideations. Another thing that I found was really interesting in Mark Hayden's talk was that psychedelics, even though they are technically illegal, were the number one predictor, more so than economic status, financial status, or like education levels. That was the number one predictor for not being incarcerated, which was very interesting. Even though they're an illegal substance, kind of says a lot about the people who consume psychedelics compared to if you look at more alcohol-related. There's definitely a lot of people in prison, incarcerated for alcohol-related situations. So in a study, 6.7% of the U.S. approximately suffers from diagnosable depression. In a study, 6 males, 6 females took about 10 milligrams of psilocybin, and then significant reduction in depression just after 2 or 3 hours and persisted 1 week later. Another study found, with a bit more volunteers, found that even 8 to 16 months later they reported more life satisfaction.
Wait, so they took 10 milligrams
pure psilocybin, though.
Yes.
Yeah.
Oh, pure psilocybin.
Pure psilocybin, and then 10 days later they took 25 milligrams.
Okay.
Not plant
So this is a cool study from Robin Carhart talking about a patient group who suffered from resistant major depressive disorder. 30% remained in remission, while overall 75% felt some relief from depression symptoms. So, I mean, that's a lot more effective for treatment-resistant depression, meaning that essentially none of these people responding to SSRIs or our current psychological treatments, 75% of them improved from psychedelic treatments. And this is a cool graph just about how the safety of psychedelic substances. Here we have the active dose divided by the lethal dose, so like the LD50 essentially, and then up here we have the dependence potential. We see that LSD and psilocybin are way down there even compared to caffeine. And then nicotine and alcohol considered our society's most common drugs. Considered society's most common drugs are way up there on, and morphine, heroin. So pretty much these are all like illegal drugs, essentially. That we're not maybe not heroin, but like
time.
Oxycodone and heroin.
Yeah, oxycodone, yeah. And many derivatives.
But then you see more like marijuana, LSD, psilocybin kind of way down on the scale. And then just to wrap things up, I really want to make a differentiation between research chemicals and classical psychedelics. There's a huge difference between them. Well, some people are willing to eat these chemicals. We don't know what they will do to you, like in the long term. Like LSD was synthesized first in the 60s, and that's been 50 years, 60 years since then. We know what will happen to you 60 years later when you take LSD. Versus some of these chemicals have been synthesized 1-2 years ago. We have no idea of the kind of effects that it will cause on you long term. So these chemicals are not safe and involve a lot of risks. And in fact, what they've been doing recently has been taking like LSD and MDMA and kind of tweaking them into more intense chemicals. And these can be fatal and very, very toxic. In fact, some chemicals can kill you in the same dose or in dosage range that LSD just wouldn't be able to. I mean, there's an arguable sense that LSD could be... Mark Hayden argued that LSD was safer than water, and his argument was that you can take 1,000 doses of LSD and live. Please don't, but you can. But he was like, what's a dose of water? A glass. You can't drink 1,000 glasses of water and live. So, I mean, it's a little bit of a stretch, but it's an interesting theory. Yeah.
And this is why test kits are important for just everyone, everywhere,
Absolutely.
no matter what.
Absolutely. And then it's kind of sad because it seems that there's generally a huge kind of fluctuation of these research chemicals with things like the Silk Road and other online sources, because some of them are not even illegal yet, so these companies can sell them online, and kids or adults will just purchase them and sell them as these substances to make quick money, because they can be very, very, very cheap. And that's why some of the dangers of just trying to do this treatment in a non-therapeutic setting, you can definitely have to be very careful about what you're taking, because especially nowadays in this current day and age, there's a lot of chemicals that are not the psychedelics that you want.
And we are going to open up just the room for any questions, and we'll end that at about 4 p.m. And then I know we have till 4:30 to kind of wrap up. So then after a quick 10 minutes of Q&A, we'll kind of mingle about and then wrap it up. So yeah, thanks everyone, and also donations cannot be accepted here today. It turns out Cedar Mill Library, we get to rent this room for free because we're a nonprofit. So just keep going out to Portland Psychedelic events and supporting the organization. We're trying to do great work and just provide the value. And yes,
How do I get more involved?
Get more involved.
You can see me.
Yeah,
Okay.
Holly, one of our organizers. Yeah.
Cool. Yeah.
Yeah. So just once again, I wanted to thank everybody for coming here, and also I wanted to say, I'm really, really glad that everyone's able to make it out to these. I'm very, very honored to be able to host the West Side, and I'm just so excited to watch what this can really turn into. I could really feel a sense of community, and I'm really excited to make friends and such. So thank you, everyone, and any questions, Aaron, please. Also, just wanted to say, also extremely grateful for Aaron. It's his birthday today, and he took time out of his
Logistics question.
Yeah.
The next meeting is.
So the next meeting's going to be in 2 weeks, and I'm working with a few people, and just so in case anyone would like to know, it's either going to be Tom and Sherry Eckert, Cherie from Oregon Psilocybin Society. They're going to come out and talk about kind of legal issues and also what OPS has got going on as far as the 2020 ballot initiative. And then also
film it because I won't be able to be there.
Oh yeah, Jason, I don't know if you'll be available.
It's not in 2 Saturdays, is it? Because we have another PTS event, 420.
Oh, that's right. Shoot.
Yeah, I mean, I'll be able to film it. Yeah, I appreciate you very much, Jason. If everyone could also give it up for Jason. I hope everyone was able to take notes, but if you weren't able to, Aaron has offered to give the PowerPoint presentation, so I'll be sending that out to everyone through Meetup, so you can go through your notes, and we'll be able to go back and source it.
Yeah, you could just take a picture of my email address, and also if you have any questions about psychedelics, I also have some other presentations as well around ayahuasca and addiction.
Yeah,
I plan to have
you can make those available to us.
I think, Aaron, you'll probably be back.
Yeah, I plan to come.
If
you want to have me back, I'd be happy to come back to talk about the ayahuasca and addiction, as well as I'd be happy to come back to talk about that psychedelic experiment with the N-Canda data, comparing psychedelic users' executive functions to non-psychedelic users' executive functions as well. So,
yeah, absolutely. So yeah, as far as logistics go, in about 2 weeks, and it'll either be Tom and Cherie, or I'm kind of thinking, since this was more on the neuroscientific side of explanation of things, maybe a more spiritual talker kind of covering that range of psychedelics, maybe just to get the other side of the spectrum, but we'll see how it goes. But thank you, everyone.
I have several questions about this study.
Okay.
Why 21?
So we need people who are as young as possible to be in the study. However,
why?
Because we're looking at developmental brains.
Oh, okay.
So we're really looking at brain development, and specifically, it's a safe, legal, non-prescription substance. So the terminology is either a stimulant such as caffeine, a depressant such as alcohol, or a painkiller such as Tylenol, or a placebo. So based on it being alcohol is one of those things, everybody in the study has to be 21. However, we're really trying to get people as young as possible, so 22 we're not interested in. So it has to makes it very hard to recruit for people.
But why right-handed?
Oh, right-handed has to do with MRI scans. So you have different, there's like ipsilateral, which means that it kind of crosses over, and so you'll have different brain regions that are kind of activated. For example, the amygdala has a left and a right hemisphere, and generally if you're left-handed, it's kind of one hemisphere is more active than the other hemisphere versus if you're right-handed. So
I do have one more question that you may not have an answer to.
Okay.
Last week, I tried Morning Glories. And had nothing. I had at least 150 seeds in there. I take Cymbalta. Would that in any way?
So Morning Glory is a chemical called LSA, which is a natural kind of component similar to LSD, but it's produced naturally. However, might I ask, where did you acquire those Morning Glory seeds?
Fair Seed Company.
Okay, a lot of times, a lot of companies, I don't exactly know about that one, but they will boil the seeds before to kind of remove any psychoactive components to it. And in fact, to produce DMT, which is a very natural chemical, there's like, you can grind up roots of Mimosa hostilis and kind of extract it from that. However, a lot of times companies will kind of boil or damage the Mimosa hostilis just to prevent any kind of people from doing that. So there's a lot of components to it that, yeah, definitely.
And you said you were looking at ADHD.
Yeah.
And I know personally, and I've had clients that are so treatment resistant that effectively they're disabled. They can't work. They can't run a schedule like, you know, most people. Like they stay up all night, maybe sleep all day, and or whatever it is. I'm just really curious if something could help them.
Absolutely. While we still need to look a little bit more into psychedelics to treat things like ADHD, I haven't really seen much research on that. There definitely is a lot of things that we can do to help them, specifically in the lab I'm working in. ADHD is like a spectrum, so you'll have some people that are more intense, less intense, some people that have completely different symptoms than the other people, like some people are more inhibition less versus other people are more attention, you know, so there's different components to it. So in my lab, we're really trying to identify what those different subtypes are, and so we can apply better specific treatment to the individual person rather than trying to make this giant bucket treatment and put everybody into it at once, because that's not going to be effective. And then, of course, you know, people who are more severe are going to need the more intense treatments, but that's a lot that we have.
When you say treatment, you mean psychedelic treatment?
Well, I'm not exactly, I haven't seen the research on ADHD for psychedelics, but that'd be very interesting to kind of look into a little bit more.
So if I contact you, you don't really have anything specific?
I can tell you a lot about ADHD, but if you contact me, I'd be happy to look up to see if there's anything on ADHD, because I'm actually really interested in that too.
Yeah.
That'd be really, really interesting.
Thank you.
Yeah.
I have a couple questions about the low-dose model that you showed when you were in.
Yeah.
It kind of looks from the imagery that you had in there like the receptors were actually being downregulated. Was I interpreting that?
Oh, are you talking about a little bit further back?
Yeah.
Oh, a little.
You have the cool little receptors that
This
popped off, that one, yeah.
Yeah.
So is that actually the receptor being downregulated?
So no, it's more, you actually didn't really see the mechanism, so I didn't have time to do the full animation. So it'd be like, you take psychedelics and then it downregulates it, and then that causes it right away.
So it is downregulated?
Yes. Low doses.
So is that psilocybin?
This was for psilocybin, yes.
So that'd be microdosing?
Yes, microdosing, yeah.
It was an actual microdosing?
Yeah, it was a smaller dose. And this is this was done in rats, post-mortem. So this is after the trip. So they gave the rats the psychedelic, they sacrificed them, then they looked at afterwards, and they found that lower dose had less activity in the hippocampus and higher dose had more activity.
Were they able to identify specifically what region of the hippocampus?
The CA. So it's the CA regions of it rather than the dentate gyrus and other regions like that.
When you're talking about microdosing, what's the threshold you're talking about?
That is a good question. Specifically, there is people who are a little bit more dose dependent, but if you're talking about mushrooms, that can be a little difficult to do because there's so much variability in the exact mushroom. However, things with LSD, you can kind of do smaller doses. So generally about, I'd say maybe 25 micrograms would be about a small dose of that, definitely.
Lower.
Or even lower, like 10, about 10 maybe.
10 to 15?
10 to 15, definitely. Yeah, I guess a full trip is about 100. So that would, that'd be kind of getting close to that.
You would get visuals probably with 25.
Probably.
With microdosing, a lot of times you don't get any of those.
I see.
You don't get any.
Depending on the strength of
Question about the people trials that you referenced.
Oh, yeah.
Are those people off of other medications when they get through that experience?
Yeah, so they were not going under any therapy, and a major component of the ethicalness of that is that these people were not responding to treatments. So we didn't ask them to stop their treatments.
Okay.
They
so
just
the medical treatments they were still on, if they were taking SSRI,
yeah,
they were
just
still on that as they went through that process.
or they were not on it because the SSRI didn't work for them, so they just stopped using it. So they weren't doing anything because they just couldn't find anything. So that's kind of a majority of the people that were in these trials.
So if an SSRI worked for somebody, if they wanted to go through some treatment with one of these other methods, would they need to go off of the SSRI in order to do that?
I would highly recommend that because the SSRI blocks the reuptake transporter. So basically, serotonin goes in the cleft, and then these little transporters suck it back up. And serotonin blocks that, which just makes more serotonin in there. And then if you already have a lot of serotonin in the cleft, and then you're adding in psychedelics on top of that, which act on the serotonin receptors, you can have way too much stimulation of the serotonin receptors and actually produce a symptom called serotonin syndrome, which can be fatal. So it's definitely good to avoid SSRIs under the influence. And this is more so for right now. We have to work with people that just don't have any kind of efficacy, or their treatments don't work for them. So they're kind of, because you can't tell somebody to stop taking a treatment ethically. So that's what we're kind of going to. But once we have more research behind it, we can say, hey, this really works. So stop your treatment and try this.
Yeah.
But once we get them a little bit more data.
How soon before the treatment would you need to stop the SSRI?
That is a good question. I would say at least a few weeks because they take 4 to 6 weeks to take effect. So you're going to want at least probably the more time is better. But I would say, again, I'm not exactly looking at the research behind this, but anecdotally, I'd say probably 4 weeks to 6 weeks.
And different ones have different half-lives. So like one you might need to stay off of longer than another depending on its half-life.
Absolutely. Well, I think we're right about out of time.
Yeah, awesome. So, once again, I mean, seriously, if you see anyone that you would like to be connected with, introduce yourself to people here. This is going to be an awesome community, and really grateful to have you all here. So, enjoy the rest of your weekend, and thank you.